If you follow how these populations are doing, they very well might give you an indication as to how other species are doing.
This is the Mojave desert tortoise that we're studying here. The Latin name is Gopherus agassizii. We've been studying in this site for about 20 years.
What we've done is that we've decoded the Mojave desert tortoise genome, and what that means is we have all the information on our computers that tell us what makes a tortoise behave the way it does, be able to survive these harsh desert conditions, but also information about what makes it susceptible to the diseases that it gets.
The genome is a very important resource for the conservation of this species. For instance, we don't really know how the tortoise is dealing with the fact that it's currently being threatened by a disease of the respiratory system. This genome will help us catalog which immunity genes are evolving quickly enough to allow the tortoise to overcome this disease threat.
Having the genome allows us to really start new collaborations that are very exciting in that they would potentially have very direct impact on saving the desert tortoises for the future. Essentially, it's the foundation for many things, including tortoise medical diagnostics, which we're doing with the U.S. Geological Survey.
In the last 100 years, we've had invasive grasses come in. They came in with livestock, probably mostly accidental. Red brome and Mediterranean grass are the two primary invasive grasses we have here. A diet of primarily red brome is really a bad diet for little tortoises. They don't grow well, and their survivorship is much lower for those that have a predominance of red brome in their diet.
We just finished up an experiment where we were looking at the effects of different diets on juvenile tortoises. So doing the transcriptome is kind of the next phase of that to really understand what genes are responding when they're stressed nutritionally and in other ways. And then, is that a marker that we could use to try to identify stress populations?
Tortoises and turtles tend to have slow mutation rates. They don't accrue a lot of changes very quickly over time, and so that can be important in terms of managing the species to know on what time scale are these species adapting or changing with their environment, because we know that we're living in a changing world, and so how are these organisms equipped or maybe not so well equipped to actually deal with those changes.
They're helping us understand the diversity, the genetic diversity of these different populations and how the animals, ultimately how that diversity helps them respond to the changes on the landscape. And that makes it very valuable information to have and very exciting to work on.